Beginners treat the abstract like a summary, but experienced readers don't. To us, it's a triage tool. In this video, I'll show you how to do the same whether you were a beginner to reading the biomedical literature or you're further on as an early career researcher, clinician, or even a fellow academic just curious how someone else does it. You're here with me, Dr. Susan Heavey, Associate Professor of Translational Medicine at UCL, and this is Further Reading. Now, this episode is the first in a little mini-series that I'm recording and sharing in a few different places. So if you're watching this first, hopefully you will see that the next video is one on introduction, because I'm going to do a video on each section of a paper as you tend to see it in a paper. So it'll go abstract, introduction, methods, results, discussion. Now, in reality, as an academic who's been around for a while, I've read, thousands. I don't really know how many abstracts. Realistically, I don't actually tend to read them in the order in which I'm putting these videos out. When I read a paper, I tend to jump from the abstract to the results figures, the results text, go back and forth around the place. And that's what you will do too once you're practiced at this. But that order is not the order that you are likely to search for videos or instructional tools on this. You're going to expect to see a series that is in the same order that the paper's written in. So that's the order in which I'm going to put out this series. But I'll clarify along the way which order I tend to actually read things in. Now, when it comes to the abstract, I do actually tend to read this first, and I read it for a very specific reason. This is something that I can do almost instantaneously, and you will too. It's purely there in front of your eyes on your screen as a triage tool. The abstract is just helping you to decide whether you're going to read the paper or not. That's it. It's that simple. If you want to glean a little bit more information from it beyond just, am I going to read this paper or not? Well, you want to probably ask yourself these 4 questions. What question are they asking? What kind of study is this? How big is it, roughly? And is it relevant to me? You kind of need those first 3 to decide whether it's relevant to you or not. And honestly, once you've been doing this for a little while, all 4 sort of happen in a weird blur of efficiency almost instantly in your head. Now, if you can't answer those questions, the abstract hasn't done its job, or maybe you're just trying to read too much into it. Okay, I want to talk explicitly to different types of researchers because for each section of the paper, and this does also hold true somewhat for the abstract, different levels of readers are going to want to get different sorts of information from it and are going to spend different amounts of time on it. So firstly, if you're a student or a beginner to reading biomedical literature, if you're, undergrad or maybe early postgraduate, your main job here is orientation. You've probably landed on this paper because it's been in a recommended reading list from a lecture. And your job is to quickly ascertain what field is the paper in, what topic does it actually sit in, has it been recommended to you because it's a good example of the lecture or the module of a whole, or is it something more specific? Maybe you're in a biomedical methods module and you're being taught PCR. You might quickly realize that this paper is just being given as an example of something that probably uses PCR in it somewhere. If it's a breast cancer clinical trial and you're studying a clinical trials module, then you're probably there because it's a good example of how a clinical trial is set up and run. If you're studying an infection or immune response module and you found yourself staring at a paper on cytokine storm in COVID-19, then you're probably there to actually understand the biology, the take-home messages that the researchers themselves were setting out to answer. So asking yourself that quick question of why am I here? Why was this paper recommended to me? And what sorts of information am I trying to glean from it is probably the most useful thing that you can do while looking at the abstract. You're not expected to be extracting any level of nuance or critiquing the methods or approach or spotting any subtle biases from the abstract alone. And if you've been taught so far in your education that you need to read papers very, very carefully and really understand any biases or limitations that might be there, really ask questions throughout, then that is fair enough. And I'm not saying not to do that. I'm more just saying that the abstract is not really the place for that. This is pretty much just Why am I here? Why has this paper been recommended to me? And what kind of things will I need to pay attention to moving forwards? Is it the methodological choices that they have made? Is it the actual biology that they are looking into? Is it the results of the clinical trial? Is it the statistics methods they used? Is it the data visualization methods they used? Is it simply an application of a technique that we've been discussing, or is it one of many examples of a biological phenotype that's been covered in these lectures or modules? By glancing through the abstract, you can see the rough shape of what's been done and know for yourself what it is that you want to read about that paper moving forward. The abstract is for you to orientate yourself and answer the question, why am I here? Now, if you're doing your first research project, maybe a dissertation or thesis project, or even your PhD, the abstract is going to help you decide, is this paper background reading so that I can get myself up to date with the literature in this field that I'm joining? Or is this something I might directly cite, maybe in the introduction or in the discussion of what I'm working on? Is this adjacent but not quite right? In which case, maybe you might glance through, but you probably don't need to in detail read this paper. Now, at this level, you should already be noticing the methods language here. Are they saying key phrases like sequencing, imaging, trials, cohort, AI? You're not necessarily judging the quality yet, but it's helping you to situate in the growing library of evidence in your mind where it is that this study sits. Now, clinicians tend to be very focused readers with a very specific question in mind when they look at the abstract. Is this likely to change how I think or practice? This usually means that the first thing you want to be paying attention to is population, outcomes, and scale. Is this a small single-center retrospective cohort? Is it a large international multi-center cluster randomized trial? Is it somewhere in between? Clinicians are alert to overclaiming, and clinicians are adept at understanding quickly how useful a paper is going to be to them before deciding whether to read it or not. And just like in their day-to-day clinical practice, this abstract is used as a triage tool. Do I need to read this paper? How urgently do I need to read this paper? And in what depth do I need to read this paper? Now, for established academics like myself, really from the level of postdoc through to PI, professor, clinical or non-clinical, you're already used to reading abstracts at speed. You're scanning quickly for things like novelty, positioning, and whether the paper is actually going to ask something interesting or something very familiar, but maybe with slightly different branding. You're seeing whether it's part of a trend in the wider literature that you've maybe noticed, hey, everybody's using this method lately, or gosh, suddenly everybody cares about that one pathway or that one class of drugs. You look at abstracts all the time. A running commentary in your mind if you supervise students and earlier career researchers is, ooh, should I share this paper with someone? So it's not just a triage tool for yourself, but a triage tool for whether you should share it with your research group, different modules that you lead, different individual collaborators that you work with. As an established academic, you're probably reading an awful lot more abstracts than earlier career researchers and students, and maybe then clinicians. And you're not necessarily reading the full paper. You're probably less likely to go ahead and read the paper in detail than a lot of those other categories of readers. That's because as an established academic, a lot of the time your job is to share the paper. Day to day, I send a lot of links, in Teams chats and WhatsApp groups and emails saying, not sure if this might be of interest. That is pretty much the extent of the message I send, and I don't know how many times I send it a day. A lot of the times that's a paper that I have scanned through the results and thought that the figures in particular looked interesting. Probably at least half the time it's something where I've only read the abstract, which is why I frame it as not sure if you might be of interest, rather than this is an amazing paper, you have to read it. If I'm saying something that effusive, I promise you it's a paper I have actually read and in detail. But largely it's purely that triage tool. Should I read this paper? Should someone else I know maybe think about reading this paper? Okay, now overall, regardless of how early you are in your reading career all the way up to how experienced you are, there is one rule that I really want you to live by. This framing of the abstract as a triage tool, it is not, do I cite this paper? It is, do I read this paper? Because if there's even the slightest chance that you're going to cite this paper, be it in an essay or coursework for a module you're studying, being it in an exam or in the introduction or discussion of your thesis at whichever level, or in the introduction, discussion, or methods or any section of a paper that you're going to publish, if there's a chance you're going to cite this paper, you need to read this paper. It's not going to be the case that you're going to look at the title and abstract and then cite it somewhere. Seriously, I mean this one. Friends, don't let friends cite papers they haven't read. The abstract just purely gets you in the door. It's a decision. It's a yes, I'll read it, no, I won't read it. It doesn't get you out of the work. Okay, I've chosen a few abstracts that we can use as examples to go through and decide whether we would read this paper or not, depending on who we are and where we're coming from, I suppose. Now, the abstracts that I've chosen are actually papers that I've covered recently in the Further Reading series. So if you do actually notice any keywords that you're interested in for your own studies or research, you can actually go watch full videos on these particular papers. So we like the efficiency route around here. These aren't random abstracts. They're ones that I've already discussed in detail. So that's available if you want it. The first paper that we're going to discuss is from Nature Cancer. It was published in January of 2026, and it's around AI in breast cancer. Now, the first thing I would do as an experienced reader, and that you should get into the habit of doing regardless of what level you're at, is noticing some keywords to help you form a quick understanding in your mind of what this paper is and what was done. So this says that it was a prospective evaluation of over 10,000 women, so a good amount of patients here. This is within one UK region, so we're not looking at a big scale international trial here. They mention AI and workflow and workload throughout the abstract, so it's clear that this is kind of to do with integration of AI into the clinical pathway. And notably, they do compare recall versus double reading. So it becomes clear quite quickly that this isn't just one of hundreds of papers that have been coming out on whether AI can work in terms of diagnosing cancer. This is actually about how it can be implemented. So a couple of different workflows that were incorporated here. They also have a headline finding near the end of the abstract that really jumps out to the reader, where they talk about having up to 36% workload savings, which is a pretty impressive claim. Now, using this abstract as a triage tool is going to depend entirely on why you're here. If this was recommended reading for a module that you're studying, then you're almost certainly going to read it anyway, but it's worth using it slightly more selectively as a triage tool to help you understand how you're going to read it. So, for example, if you've come here from a general cancer module or a breast cancer module, then probably this paper is here as an example of something that's happening lately in cancer in general, which is looking at AI to help reduce workload in terms of diagnosing breast cancer, as well as, of course, other cancers as well. So this is probably going to be one of those papers that you're to read as just an example. What's happening? With that in mind, you, yes, should go ahead and read it. You don't need to necessarily do so in a huge amount of detail unless it's a paper that's, been really pointed out by the educator as something that's a really useful one. It picks up on different points being made in your module. Other than that, you probably just need to read through and have a basic understanding of okay, this is the kind of thing that's being done in this space at the moment. However, if you've landed here through a module that's maybe on something like AI, you can see straight away from the abstract that this is pretty sophisticated. You probably already know enough about AI integration in clinical workflows to know that this is a better paper than a lot of the ones that are coming out here because it's prospective rather than retrospective. They did a couple of different workflows and compared them. It's also a decent amount of women, not huge. It is within one region. It's not international, but it's still a pretty good paper. It also doesn't harm things that it's in a pretty good journal, Nature Cancer. So if you're here because it's been recommended to you from something AI specific, then this abstract is enough for you to say, okay, hang on, this looks like a good read. This is a read that I should dedicate some time to and go into in a bit of detail. I should understand the exact types of AI that were used, the different ways that they were incorporated into the workflow, and I should critically assess the claims that were made about efficacy here. If you're reading this paper as an early career researcher who's working in a topic directly relevant to this, then yeah, you can see from these keywords that we've already discussed that this paper is worth your time reading as background reading with a view to probably including it in an introduction or discussion, depending on how relevant it is to your specific research topic. And from looking at those keywords, you can probably already decide whether there's a decent chance of you citing it or not. If you're in a similar but slightly adjacent research topic, like say, I don't know, for example, maybe you're studying new treatments for breast cancer, then you can see from this abstract that it's pretty cool, and so it's worth knowing. It might even be something that on a tangent you end up discussing in lab meeting or maybe even in your Viva. It's worth knowing that this is something that's happening in the diagnosis space and worth considering how it could be applied to the treatment space, but it's probably not directly relevant to your specific study. You're probably not likely to formally cite it, but it probably is worth having a glance through. If you're a clinician, this paper is going to be of interest if you work in, for example, referring patients for suspicion of breast cancer, to know that this is the kind of thing that's coming on board in hospitals and to be aware of it. But of course, if you're actually working in the diagnosis space for breast cancer, then you're going to know right from the get-go, probably even from the title, that this is something that could affect your clinical practice. It's something that might help you to consider any AIs that are already being incorporated in your clinical setting, whether maybe the workflow that you use around those is optimal or maybe potentially is something that could be tweaked in view of what was found here. And with that in mind, you're certainly going to want to read this one with a critical lens and really consider it carefully. And for me as an academic, I'm usually glancing at this and saying, okay, which modules might this be a useful read for their recommended reading? Which members of my research team might find this interesting for their background reading? Is there any members of my research team or collaborators who might want to cite this paper in their research? And that's who I'm going to share them with. Okay, the next paper that we use as an example for how to read their abstract is this one from Nature Biomedical Engineering that was published in January 2026. And again, we had a recent further reading episode on this if you're interested in watching that. Now, the title already gives you the model, the organ, the cell type, and the disease context. So that's already going a long way to help you with triage. The abstract itself looks pretty dense. There's a lot of jargon and keywords that if you're a beginner to this field will already feel potentially a bit overwhelming, but try to use them to triage how deep you want to read this paper. The abstract tells you the model system, that's patient-derived human organoids, into multi-regional assembloids. The biological focus is parietal cells and gastric acid secretion. The big question is, can organoids recapitulate function, not just structure? So this is a methods-driven primary research paper. It's really mostly about the model itself as opposed to findings from it, although of course those are included given it's a high-impact paper. If you're learning about stem cells, differentiation, or epithelial biology, this is highly relevant, but it's even more relevant if you're learning about new model systems and better ways to study disease in the lab environment. If you're revising for exams, this paper is going to be useful for examples, not for memorizing the deep details. In terms of triage, as a student, you're going to want to read this selectively, probably looking at the figures and maybe the introduction. It's not going to be essential to look into the specific types of cells and the mechanistic biology unless the module is actually covering GI biology. And if it's just been given as a light example of some cool new methodologies in a broad cancer module, then you can do a light read just saying, look, this appears to be something that's happening lately. People are using patient-derived models that can self-assemble and show different regions and different phenotypes. If you're an early career researcher, usually a PhD or postdoc, then the keywords that immediately are going to jump out to you are around the self-assembling aspect into these multi-regional assembloids as opposed to organoids. You're also going to see that they're looking at functional responses to drugs. You're going to clearly see that it's patient-specific modeling, which makes it both highly relevant to individual patients and perhaps poorly applicable across large cohorts. The thing that should jump out to you really quickly is that this is a platform paper. It's not just a biology finding. It has further relevance and could be incorporated into wider and more important studies as time goes on. This is something that can have really important downstream impact, not just for patients, but for researchers. So we've got stacking functions here. Now, if you're closely in this field, then you would already know that claiming emergent function, so in this case, parietal cell maturation, is something that's notoriously hard to do in vitro. So you're going to notice right off the bat that this is an interesting paper that you should, yes, go ahead and read. And you're already getting a sense from the abstract that the disease modeling is more of a proof of concept. It's not the main technical contribution. So in terms of triage, yes, you would go ahead and read this if you're working in organoid research, assembloid research, 3D models, or things like epithelial crosstalk or translational disease modeling in general. And you're going to skim it more lightly if your work is purely computational, or maybe you only work in the immune area or non-epithelial systems. If you're a clinician triaging this paper, then straight off the bat, you're going to notice that this is quite a technical lab-based paper. You're going to see that they're patient-derived models and that they are from a cohort that is pediatric. It's a genetic condition. They've got impaired gastric acid secretion, and that they're looking at functional response to drugs. But it's not a clinical trial. It's not proposing a new therapy. And although it does look interesting, it's very science heavy. So it's going to be perhaps a bit more of a laborious read to catch yourself up on the lab jargon. It's clear that this is a paper about mechanism and modeling, and it's not something that's going to immediately change your practice in the clinic. So from a triage point of view, you're much more likely to move on to the next paper. That being said, if you work in rare gastric disorders, mechanism-based understanding, or if you have close collaborations and are involved in research yourself around translational modeling, then yes, of course you might give it a read. And if you're a more senior academic or a PI, then you're going to notice the key here is about the model itself. It's platform building, and you should be considering whether this is a platform that you might want to incorporate down the line in any of your main projects or that your collaborators might be looking into. It's a methods forward paper. It's got broad reuse potential, and so it is likely something that you're going to read yourself and contemplate how useful it might be to future grants. That is if you're working in a relevant space. Now, the key here, I think, no matter what level you're at, is that part of this triage is going to be that this is a kind of fairly dense abstract. It's got a lot of specifics here around the methodology. And so if you're deciding that, yes, you're going to read it, you're probably going to be committing a bit of time to it to catch yourself up, unless you're exactly right within this very tight field. Here's an example from another recent video. This is from a paper published in the Lancet in February 2026, and it's a good example of a structured abstract, which you'll often see in journals like this. Not every journal does it, but when they do, it does help you to triage much faster. So this abstract is broken into background, methods, findings, and interpretation. I want to normalize that format does not equal quality, but format does help speed. If you're a student, you're probably looking at this paper as recommended reading from part of a module or program on public health, epidemiology, or biomedical sciences. And what you should look at first are the background and method sections. This will tell you immediately that this is an observational study. It's not interventional. It's got multiple cohorts, means that it's not a single dataset result. And global burden estimates tell you that there's a modeling step later. So if you're learning about risk factors, cohort studies, or population health, this is relevant background reading, and you should go into it in a little bit more detail. But you don't need to interrogate things like the causal language at the abstract stage. Just use this as a, yep, I should go ahead and read it. Now, if you're an early career researcher, PhD, postdoc, the key signals that you're going to get from the abstract are, again, that it's multi-cohort. The outcome definition is severe infections. and that they are looking at global burden estimates. So there's a secondary modeling layer here. So it seems this research is doing two things. They're estimating association, then scaling the impact. And those are analytically distinct steps. I'm already noticing that. The abstract lets me separate primary evidence from modeling. If your research is on something like obesity or infection risk, maybe non-communicable disease interactions or global health modeling, then yep, it's going to be worth reading for you. If you're a clinician, this is something you're already going to be pretty well practiced at reading. The Lancet is going to be a familiar place for you. And you're going to notice probably straight away that this paper is not about individual patient risk. It's about population level association. You're also going to notice straight away that the outcome here is severe infection, not infection incidence. And you're going to be aware that global burden estimates are about scale, not bedside decision making. So it's not necessarily going to impact your clinical decision-making tomorrow. It's more going to help you think, okay, how big is the problem? A little bit more background reading on where we're at in terms of obesity research and particularly its relation to severe infection. So you should read it if you're interested in prevention, risk stratification and discussions, public health context. But you're already going to know from the abstract that it's not a guideline paper, it's not practice changing, and it's not something you need to prioritize to read right this second before you see your next patient. For a more senior reader or an academic, you're going to extract key information straight away in under 30 seconds. You're going to see the exposure is obesity. You're going to see the design is that there's multiple cohorts, so it's robust. The outcome is severe infections, so it's got healthcare relevance. And you're going to see that add-on of the global burden around policy framing. It might be useful in introductions or discussions of theses that you're supervising. It might also be useful in public health framing. And if you're teaching modules around things like clinical trial design, clinical study analysis, it's going to help you with all of those kind of key examples of things like correlation does not equal causation and so forth. Hopefully those few examples helped you to understand where at your level you should be kind of focusing your energy on an abstract. It's not for understanding the whole paper. It's purely just for deciding what kind of paper this is and whether it earns more of your time or whether the next 10 or 30 minutes or maybe even an hour might be better spent elsewhere. Now, wherever you're watching this, it is part of a series, and so you can see it in a playlist or a course in whichever website you're at. So we will be looking at how to read an abstract, how to read introduction, methods, results, and discussion, depending on what angle you're coming from. In each of them, we'll also use examples from other further reading videos. And following on from this series, a great place to continue would be to look at some of the videos of specific recent papers that have come out. Whether they're part of your field or others, they help to show you how you can read a paper, as well as some of the broader concepts around paper craft. That's how to write a good paper, how to use interesting data viz, and how to get the most out of reading it. So hopefully this is the start of a closed loop for you that will help you to learn how to read and ultimately write better biomedical research. If this was useful, please let me know. There's still probably time to tweak future episodes in this mini-series. And if not, well, the next mini-series will be better for it. So let me know. Does it align with what you're already doing? Does it align with what you're currently being taught? Does it align with how you teach others? If you've made it this far, well done. Happy reading, and see you next time.